In this video, we are going to learn about the endomembrane system. The endomembrane system is a network of interconnected membranes, and they are connected either physically or by vesicles. So this includes the nuclear membrane, which is right here, and it is physically connected to the endoplasmic reticulum, which is all these folded membranes outside of it. The endoplasmic reticulum comes in two different forms, smooth and rough. Smooth lacks ribosomes on its surface, whereas rough endoplasmic reticulum has ribosomes on its surface. And remember, ribosomes are what produce proteins. All right, next up, you will see that we have kind of some of these ball-like structures. Those are going to be vesicles that are going to help connect and transport kind of membranes and materials between these organelles. And we also have the Golgi apparatus, which you can see are these folded membranes right here. Okay, the Golgi apparatus's job is to kind of receive proteins. It will then modify those proteins and then put them in vesicles. And then last, we have the cell membrane, which is part of the endomembrane system. And the cool thing about this is because of the fact that membranes are made of phospholipid bilayers, okay, they're very flexible. And we know that the tails of those phospholipids are hydrophobic and the heads are hydrophilic, okay? So those hydrophobic tails want to stay together. And so basically you can see that these kind of vesicles and things like that can kind of fuse with these membranes because of the fact that these hydrophobic tails want to be dry and with other hydrophobic tails. And so that's going to allow this kind of movement between them. All right, so let's walk through exactly how the endomembrane system works. It has two main jobs. It can produce and modify molecules to be exported to other parts of the organism, which is this pathway right here that we will walk through. Okay, another job that it can do is it can break down toxic chemicals or substances within the cell, which is what we're seeing here in this pathway. So we're going to walk through both those pathways to help us understand that. All right, so let's start off with a protein-based secretion from the cell. So what are some examples of protein-based secretions? We will learn lots more about them later on, but there are protein-based hormones which are able to travel, say, through the bloodstream to other parts of the body where they will bind and start off signal transduction pathways, or kind of these communicative pathways that allow for the receiving cell to do a different function. You might also see things like antibodies. So in white blood cells, specifically B cells, they produce these special Y-shaped proteins which are released into body fluids, and they can bind to foreign particles such as maybe bacteria or viruses that might be infecting the individual. So there's a lot of different protein-based secretions that you can have. So let's start off, and we're going to kind of number our way through. So we're going to start with number 1 in the nucleus. And then we are going to go to number 2 in the rough ER. We're going to go to number 3 with the vesicle, number 4 with the Golgi apparatus, number 5 with the vesicle, and number 6. Okay, so let's walk through those 6 steps. All right, so let's start off in the nucleus. Okay, remember the nucleus has DNA. Okay, the DNA is large. It is not able to leave the nucleus through nuclear pores. So it contains the directions for making a protein. However, it cannot leave. So there has to be a process that can take the code in DNA and rewrite it in a smaller molecule that is able to leave. And that smaller molecule is messenger RNA. And the process that is occurring is something that's called transcription. So transcription is when you take the code in DNA and you rewrite it as messenger RNA. The messenger RNA is then able to leave through a nuclear pore, and it will enter the rough endoplasmic reticulum. The rough endoplasmic reticulum Okay, gets its name because it looks rough because it is studded with ribosomes. Ribosomes are the site of protein synthesis. So I'm going to change my color so we can kind of see the next step for number 2. Okay, at the ribosomes, that messenger RNA will be read and a protein will be produced. Okay, so when you go from RNA, messenger RNA, to protein, that process is called translation, and that is occurring at these rough endoplasmic reticulum. All right, so also in the rough endoplasmic reticulum, there may be chaperonins, which help fold proteins properly. But in the end, you're going to end up with a folded protein. So it may travel through the ER and then be folded up. And it will basically bud off of the rough endoplasmic reticulum in a vesicle, which is what you can see here. Okay, so our next one is going to be a transport vesicle. So a transport vesicle is basically just a membrane sac that contains smaller molecules, in this case, proteins, that we are then going to travel, and it's going to attach to number 4, which is the Golgi apparatus. Okay, so the Golgi apparatus right here, okay, is going to receive that transport vesicle. And so you can see here that basically this vesicle, because it's a membrane, can kind of fuse with the membranes of the Golgi apparatus, and the protein contents inside can be released. And this can be where our polypeptide is then joined with other folded polypeptides to produce a protein with quaternary structure. This may be a place where a lipid is added to a protein to make something called a lipoprotein, or maybe you have a type of carbohydrate added to a protein and you have something called a glycoprotein. But basically it's going to modify these proteins, and then once they're modified, you'll see that those proteins are going to bud off in yet another vesicle. So we're going to go back to step number 5, which is yet again another transport vesicle. Okay, and in this case, because we're secreting it from the cell, that vesicle will then fuse with the cell membrane, and through a process called exocytosis, it will release the contents to the fluids around it. Okay, so we have nucleus with transcription, we have rough endoplasmic reticulum with translation, we have a vesicle for transport, we have Golgi for modification, we have another transport vesicle for transport, and then we have the cell membrane for the process of exocytosis. Now, I do want to let you know that although we talked about secreting proteins, a very similar pathway can happen with a protein that may be embedded in the cell membrane. So maybe we're trying to add some more receptors to our cell. So you can see a similar thing here. So we would start off with the nucleus, we would go to the ER, then we have a transport vesicle. Notice though that our protein is basically embedded in the vesicle membrane, okay? And then it will travel through the Golgi apparatus and again into a transport vesicle, and again it's embedded in the membrane, so that when that vesicle fuses, now that protein is part of the membrane rather than being secreted. So that's just another way you can kind of get that same effect. Now, another pathway that this endomembrane system can produce is something called a lysosome. So think back to lyse, like hydrolysis. So we're cutting, we're slicing, we're digesting materials. Okay, so that is what's happening inside a lysosome. It contains hydrolytic enzymes that are able to digest materials. Now, not shown in this picture, you would also have the nucleus, which would contain the DNA, okay, and would be the site of transcription. And then you would have translation at the rough ER. You would see here we have this transport vesicle containing the enzymes that we've made. So that is the key thing. Back here, we were making a protein that was going to get released. So that protein that was made was one that was going to act somewhere else. In this case, when we're making a lysosome, the protein that's inside the vesicle is the enzymes that are going to do the digestion. Okay, that transport vesicle goes to the Golgi, and then any modification to that enzyme will occur. And then basically, instead of going to the membrane, that vesicle that's released is a lysosome, and it can go ahead and do its job. So here you can see this lysosome is fusing with a vacuole-containing food, and you can see that the enzymes are then digesting the food, breaking it down into smaller pieces. Or here you can see the lysosome is fusing with, it looks like maybe a broken or damaged mitochondria, and it's breaking it down as well. Okay, so in this case, it's just a matter of what is the function of that protein that's being produced in the rough ER? Is it going to be an enzyme that needs to be in the lysosome, or is it one that needs to be part of a membrane, or is it something that needs to be secreted? So we see all of these endomembrane systems kind of working together to try to produce these proteins. One thing I do want to say is I keep talking about the rough ER having ribosomes and making proteins. I do want you to know as well that there are free ribosomes. And free ribosomes are going to deal with the messenger RNA for proteins that are directly floating around in the cytoplasm. So not ones that are involved with membranes at all. So don't forget that there are ribosomes in the cytoplasm, but whatever protein products are made, they're not packaged up into any type of membrane. They're just going to be proteins that are kind of working in the cytoplasm directly. All right. Well, I hope that helps you understand how these organelles work together to keep our cells working and alive.